Welcome back to the deep dive. Today, we're getting into something really critical. The urgent need for much faster, much more precise diagnosis of acute leukemia.
Yeah, AL. It's an incredibly aggressive blood cancer.
Affects both adults and children. And, when someone gets that diagnosis, every single hour counts. Getting the right treatment started fast is paramount.
That urgency, that's really the core problem we face with the current system, isn't it? Acute leukemia, AL, it demands immediate, critical decisions about treatment.
Right.
But the standard of care, what we call SOC, well, it's just notoriously slow. We're relying on a whole battery of tests.
Like what kinds of tests?
Things like flow cytometry, which looks at the markers on the surface of the cells. Then there's karyotyping for looking at big changes in the chromosomes, and targeted gene sequencing.
Okay.
But pulling all that data together, getting a complete picture, that often takes days, sometimes even weeks.
Days or weeks, when time is literally life or death.
Exactly. It's a huge delay in planning critical treatment. And, besides just the speed issue, SOC often doesn't capture the full complexity of AL.
How so?
Well, leukemia is incredibly varied. It's heterogeneous, lots of different subtypes driven by different molecular problems. And if you miss identifying the specific driver, you might end up starting the patient on the wrong track, treatment-wise.
Okay, so the old way is slow, sometimes misses the full picture.
Yeah.
What's the innovation? What's the potential game changer we're diving into today?
We're moving beyond just looking at the DNA sequence, the basic blueprint,
Right.
to something called DNA methylation profiling.
Methylation. Okay, can you break that down a bit? What is that exactly?
Sure. So think of the DNA as the cell's blueprint, the instruction manual. DNA methylation, that's like a layer of sticky notes or instruction markers on the blueprint.
Okay.
These markers tell the cell which parts of the blueprint to actually read and use and which parts to ignore or silence. It controls the cell's epigenetic state, how the genes are expressed, not just what genes are there.
Epigenetics. I've heard that term. Has this been used elsewhere in medicine?
Oh, absolutely. This approach, DNA methylation profiling, it's already made huge waves in neuro-oncology. It's actually now part of the official WHO World Health Organization classification for many brain and central nervous system tumors. It's that powerful.
Wow. Okay, so we're taking this proven epigenetics approach and combining it with some serious tech to get that speed right.
Exactly. Two key pieces of technology make this possible. First, nanopore sequencing. This lets us read those DNA methylation patterns in real time as the sequencing happens.
Real time, so not waiting days for a batch run.
Correct. And second, we have machine learning, specifically a neural network, an AI that's been trained to interpret this complex methylation data basically instantly.
So our mission today is really to unpack this new framework. Rapid epigenomic classification of AL using this AI engine. What's it called again?
It's called MARLIN. Stands for Methylation and AI-Guided Rapid Leukemia Subtype Inference.
MARLIN. Got it. Okay, let's start at the beginning. To build any AI that can classify something complex like leukemia, you need a really solid foundation, right? A map. Tell us about the reference map they built for MARLIN.
Absolutely. You can't teach an AI without a good textbook, essentially. They created what they call the Acute Leukemia Methylation Reference. And this wasn't just, a handful of samples. No, they did a massive aggregation, pulled together data from 11 different published studies, curating 2,540 high-quality AL samples, a huge effort.
2,500 samples. That sounds like a truly comprehensive data set. What kinds of patients were included?
It's the largest methylation blueprint for AL ever put together.
Okay.
It covered both kids, 1,838 pediatric samples, and adults, 702 samples. And crucially, it included all the major types of acute leukemia, AML, acute myeloid leukemia,
Okay.
BAL, B-cell acute lymphoblastic leukemia, TAL, T-cell AL, and also the rare, trickier ones like ALL, which is acute leukemia of ambiguous lineage.
So a really broad spectrum. And looking at this huge data set through the lens of methylation, what did it reveal? Did it change how we see leukemia subtypes?
Oh, dramatically. It showed just how much variation, how much heterogeneity we were previously missing. Using methylation patterns, they defined 38 distinct classes across all those lineages.
38. How does that compare to what we knew before?
Well, the breakdown is 21 classes just within AML, 11 in BAL, 5 in TAL, 1 specific to ALL, plus controls. But the really striking thing, 13 of those 38 classes were completely novel.
13 new types.
Essentially, yes. 8 new subtypes within AML, 1 new one in ALL. Methylation patterns revealed distinctions that standard methods just hadn't picked up. It's like discovering 13 new roads on a map you thought you knew.
That's incredible detail. But let's circle back to something you mentioned. standard tests, SOC. They're not always wrong, right? The study showed pretty high agreement, didn't it? Like 97%.
That's right. 97.3% concordance overall between methylation classification and the traditional lineage assignment based on immunophenotype, which is good. It shows methylation aligns well most of the time.
So it's 97% concordant. Why do we need this new tool? Where's the real value add?
It's in that remaining 2.7%, those lineage discordant cases. That small percentage represents the really difficult, the ambiguous leukemias where the standard tests struggle or even fail. That's where methylation really shines.
Okay.
It cuts through the ambiguity in those tough calls. That's the core value proposition.
You give an example of that ambiguity. Why would looking at methylation be better than, say, looking at the cell surface markers with flow cytometry?
Well, immunophenotyping looks at the surface markers, basically, what the cell looks like on the outside. Methylation looks deeper. At the epigenetic instructions, it reflects the underlying molecular engine driving the cancer. Yeah. And in those tricky discordant cases, over 81% of them, the methylation profile actually matched up with a known molecular alteration that often causes lineage confusion. Things like BCL11B activated ALL, or HOXA9 activated TLL, or ZNF384 rearranged BAL. These are known tricky actors.
So let me see if I understand this. Flow cytometry might say, this looks kind of like BL, but maybe TL, we're not sure. But MARLIN, looking at the methylation, could say, hold on, the epigenetic signature is unmistakably this specific type, driven by this known rearrangement, even if the surface markers are confusing.
That's exactly it. It identifies the root molecular cause, which is ultimately the most critical piece of information needed to choose the right therapy. It gets to the why behind the leukemia.
Let's zoom in a bit now, maybe focus on AML, acute myeloid leukemia. You said this analysis had, almost 1,300 AML samples. That's double previous methylation studies. What new insights did we get specifically about AML?
That scale really allowed for refining things, especially around risk stratification. We know many AML subtypes are defined by specific gene mutations, like PML, RRA fusion, or CEPA mutations.
Right, those are known genetic markers.
Yes. But methylation adds another layer. It helps us understand the risk associated with those genetic markers even better. It's not just if the mutation is there, but how the cell is epigenetically programmed around it.
Okay, can you give us a concrete example? How does methylation refine risk when we already know the gene mutation? You mentioned CEPA.
CP is a perfect example. Generally, having a CEPA mutation in AML is considered favorable, associated with better outcomes.
Hey.
But this study found something interesting. Some patients had the CEPA mutation genetically, but their methylation profile didn't fit into the expected CEPA methylation class. They were epigenetic outliers.
Outliers, why?
Well, often these outlier cases either lacked the specific type of CBAM mutation, usually linked to good outcomes, the BZIP in-frame indels, or they had other competing mutations known to be aggressive, like NPM1 or IDH12 mutations, overriding the CBA effect.
So genetically, they might look okay on the surface, but the methylation pattern revealed something else was going on. A different set of instructions was running.
Precisely. And crucially, did these epigenetic outliers have worse outcomes?
Did they?
Yes, they did. Significantly worse survival, both in the adult patients and the pediatric ones. The log rank p-values were quite stark. It really shows that just knowing the mutation status isn't the full story. Methylation tells you if that mutation is actually driving the bus, epigenetically speaking, or if something more sinister has taken the wheel.
Yeah, that's fascinating. It adds so much nuance. Now, let's talk about something else really exciting from the AML analysis.
Yeah.
These defined HOX-activated subgroups. Why is knowing about HOX activation such a big deal in leukemia right now?
It's huge because of a new class of drugs emerging, menin inhibitor.
Menin inhibitors, okay.
Yeah. These are targeted therapies, and they seem to be highly effective against leukemias that are driven by the overexpression of certain genes called HOXA and HOXB genes. This overexpression is really common in leukemias with specific genetic drivers like rearrangements in the KMT2A gene or mutations in NPM1.
So if you know the leukemia is HOX activated, you might have a very specific, effective drug available.
Potentially, yes. That's the hope in the direction things are moving. And what the researchers did here was identify 9 distinct AML methylation classes that are specifically characterized by high expression of HOXA9 and HOXB5.
9 different HOX-related methylation groups within AML.
Right. And what's really powerful and where methylation profiling truly excels is finding this HOX activation signature even when the usual genetic suspects like KMT2A or NPM1 aren't obviously mutated or rearranged.
So it can find the HOX signal even if the genetic cause is hidden or maybe unknown?
Exactly. That's maybe one of the most clinically relevant findings. They looked at one specific group called HOX group 4. Almost a quarter of the samples in that group, 72 cases, had no known driver mutation found by standard genetic tests.
Wow, so genetically unsolved cases?
Correct. But over 95% of those genetically unexplained cases showed a strong, clear epigenetic signature of HOXA9 and HOXB5 activation.
So methylation becomes a proxy, a way to see the functional outcome, the HOX activation, even without knowing the precise genetic trigger.
Precisely. It means methylation profiling could be a fantastic screening tool. It can instantly flag patients whose leukemia is functionally HOX-driven, making them potential candidates for menin inhibitors, even if their standard genetic report comes back ambiguous or negative for the usual drivers.
That could open up treatment options for patients who might otherwise miss out. And did this methylation subtyping also refine prognosis within known high-risk groups, like those with KMT2A rearrangements?
Yes, it did. KMT2A rearranged AML is generally considered a poor prognosis group. But when they stratified these patients based on their specific HOX methylation class, groups 5 through 9, they saw really significant survival differences.
Like what?
For instance, patients falling into group 6, which is often associated with KTA6A rearrangements, and group 5, linked to MECOM activation, had particularly poor survival. Median overall survival was sometimes less than a year, or just around 2 years.
Really grim.
Very. But contrast that with group 9, linked to FOXC1 activation, those patients showed comparatively better outcomes within the KMT2A rearranged cohort. This kind of substratification is just essential for tailoring treatment intensity and exploring novel approaches.
Okay, incredible layers of detail there. Let's shift gears slightly and talk about the engine driving the speed, Marlin. We know it's a neural network using this methylation data. What makes it tick, especially since it needs to work with data coming off that nanopore sequencer in real time, which you said starts sparse?
Right, the real-time aspect is key, and that's where the machine learning engineering gets really clever. Nanopore sequencing gives you data bit by bit almost immediately. So when you start a run, the methylation data you have is very incomplete, very sparse.
Like trying to recognize a face from just a few pixels at first.
That's a great analogy. MARLIN is trained on a huge number of potential data points, over 350,000 specific locations on the DNA called CPG sites where methylation can occur. To make sure it could work reliably, even with very few of those points measured early on, they trained it under brutal conditions.
Brutal how?
They used a massive dropout rate during training. In each training cycle, they randomly ignored 99% of the input CPG data.
Wait, 99% dropout? They trained the AI to make a diagnosis using only 1% of the potential information at any given time. That sounds almost impossible.
It sounds counterintuitive, but it forces the neural network to become incredibly efficient. It learns to focus only on the absolute most informative methylation sites, the ones that give the biggest clues, even when most of the data is missing.
So it learns to work well in the dark, essentially.
Exactly. It ensures the model is robust and accurate, even when the sequencing has only been running for a short time and the data coverage is low.
Okay, so the training was tough. How did it actually perform? Did it work reliably under those sparse conditions? What do the metrics say?
The performance was really quite exceptional. They used something called the F1 score to measure accuracy. Think of it like a combined measure of precision and recall, where 1.0 is perfect.
Right.
Even when simulating extreme sparsity, down to only 3% of the CPG sites being covered, that's about 10,700 sites instead of 350,000. MARLIN maintained a median F1 score of 0.91 for predicting the specific methylation class.
Wow. 0.91 accuracy with only 3% of the data. That's impressive.
And for predicting the broader lineage, like AML versus BAL, the F1 score was even higher, a median of 0.99. Super reliable even with minimal data. They also set a high confidence threshold for clinical use. The AI has to be at least 80% confident, a score 0.8, before issuing a prediction.
That robustness is key if you're going to use it clinically. Which brings us to the real-world impact. Let's talk about those real-time patient cases. This is where the speed advantage really hits home, right? Tell us about case RTC001.
Right. This was a 61-year-old woman with suspected acute leukemia. Using the standard diagnostic pathway, the full workup took 4 days to confirm AML with specific features mutated TP53 and aneuploidy.
4 days, okay, and with MARLIN.
With MARLIN, running on the nanopore data. They got the exact same classification AML, TP53 aneuploidy enriched class, after just 40 minutes of sequencing time.
40 minutes.
40 minutes of machine running. The total turnaround time from when the sample arrived in the lab to getting that definitive classification was just 96 minutes.
4 days versus 96 minutes. That's astounding. That's the difference between someone waiting anxiously for nearly a week, delaying treatment decisions.
And getting a clear diagnosis and potentially starting targeted planning before lunch on the day they came in, that's the potential clinical reality.
Let's look at another one. Case RTC002.
Similar story. A 62-year-old man. Conventional diagnosis took 3 days to confirm AML with a specific set of mutations: NPM1, TT2, and FLT3-ITD.
3 days. And MARLIN.
Again, after just 40 minutes of sequencing, MARLIN provided the classification. AML HOXA-B activated group 2, which is known to be enriched for exactly those mutations like NPM1 and TT2. So again, rapid confirmation consistent with the later, slower genetic results.
It's not just faster, though. The sources mentioned MARLIN also helped clarify things the standard methods missed, like a diagnostic blind spot.
Yes, absolutely. There was a retrospective case, AL002. Initially using standard methods, this patient was diagnosed with B-ALL, not otherwise specified. Basically, it's B-cell leukemia, but we don't know the specific molecular driver. Kind of an ambiguous bucket.
Okay.
But when they ran the methylation data through MARLIN, it assigned a perfect score, 1.00, to the B-ALL DUX4 rearranged methylation class.
DUX4, what's that?
DUX4 rearrangements are a known important subtype of B-ALL with specific prognostic implications. It's often cryptic, meaning standard tests like karyotyping can easily miss it.
So MARLIN flagged it instantly.
Instantly and with perfect confidence. And later, more in-depth sequencing confirmed that, yes, there was indeed a hidden DUX4 translocation that the initial SOC workup had completely missed. MARLIN turned an ambiguous diagnosis into a precise, actionable one.
Wow. Okay, so putting this all together, what's the big picture here for diagnosing acute leukemia? Is MARLIN meant to completely replace the old tests?
I wouldn't say replace, not yet anyway. It's more about fundamentally complementing and enhancing the current system. Think of it as a powerful new tool in the toolbox.
How does it complement?
Well, first, it offers that incredibly rapid molecular classification, often within just a couple of hours of the sample arriving. That speed is transformative. Second, it resolves those critical blind spots, the cryptic rearrangements, the lineage ambiguous cases where traditional methods often struggle.
Right.
And third, crucially, it provides these predictive signatures, like the HOX activation status we talked about, which can directly inform therapy choices, sometimes irrespective of the specific underlying genetic mutation.
And technologically, you mentioned nanopore sequencing in the AI. Is this something that's feasible for hospitals to actually implement, or is it super complex and expensive?
The researchers describe the framework, the nanopore sequencer, coupled with the MARLIN software as being relatively simple and potentially less expensive to implement compared to the whole battery of traditional tests.
That's huge.
It is. It opens the door to making this kind of rapid, comprehensive diagnosis more accessible, potentially even in lower-resourced clinical settings around the world. It could help level the playing field for leukemia diagnosis globally.
So it's this elegant blend of cutting-edge epigenetics, smart AI, and real-time sequencing technology that's really poised to redefine the timeline for cancer diagnosis.
Exactly. It compresses days or weeks of investigation into maybe 2 hours.
Okay, so we've seen how MARLIN connects the epigenetic dots to known molecular types, refines risk stratification like with SEPA, and crucially, gives almost instant confirmation of therapeutically relevant groups, like those HOX-activated leukemias. This really brings us to a fascinating point to leave our listeners with.
Yeah.
Considering this power, especially the ability to rapidly classify HOX-activated subgroups, how might this directly change first-line treatment protocols in the near future? If a doctor knows for sure in under 2 hours that a patient's leukemia is HOX-driven, could we see a future where maybe they skip some traditional chemo initially and go straight to targeted therapies, like those menin inhibitors?
That's the million-dollar question, isn't it? The clinical trials exploring exactly that the optimal sequencing and combination of these targeted agents with standard therapy are already happening. The technology, as we've discussed, seems ready for that kind of paradigm shift. Whether the clinical evidence will support using it up front like that, well, that's what we need to find out.
A really compelling thought for the future of leukemia care. Thank you so much for walking us through this deep dive today.
My pleasure. It's certainly an exciting development.